So Oxford University, from there a technology spinned out, and that was called as Oxford Nanopore sequencing technology, which claims to be the fastest and they claim to have the smallest sequencers, but still it is in the process of development. If we look into the cost, they claim that they can sequence a genome in 15 minutes with a cost which is less than $1,000, and it is expected to be available by the end of this year. So if we could get it, obviously that will be the technology of future. This is how it works. So what happens here is we see there are some membranes, and through those membranes there are some pores which are some nanometer sizes. So those pores, there is an electric current which is flowing through them. So we let some molecules pass through, and those nucleotides, when they pass through, they actually change the flow of current. So right now the current is steady and an ion is passing through, and we see that in the current there is actually a change in its amplitude. Another molecule is passing, so which changes this amplitude differentially. So in this way we can detect that which molecule is there by comparing the intensity of change in this electrical signal. So here we see this Oxford Nanopore system in detail. So what happens is that the DNA, it can be sequenced by threading it through a microscopic pore in a membrane. So this membrane is right here, and there is a pore in it, and this membrane is then we see that there are two proteins. So there is one protein at the top. So the DNA comes and enters through this protein, and it becomes single stranded. Okay, and then the second protein, that contains an adapter molecule, so that keeps those bases long enough so that they should pass one by one and detected. What happens next is we allow the ions to move through this pore. So then the electric current is already passing, and since these nucleotides are going inside, so they change the polarity of those ions, and then they generate the electrical signal, so which is then detected. So different nucleotides, they change in the current differentially, so we can detect which base is there. So we have seen that Oxford Nanopore sequencing is based upon sequencing via change in electric pulse or current. That is due to the interaction of the ions with the bases of the DNA, and this happens in the small nanometer sizes pores. That's why we call them as nanopore sequencing technologies.